by Maria Lopes. She's based at the University of Minho, Portugal, and she will talk with us about early life skin and intestinal Staphylococcus epidermidis phages, the prevalence, the characterization, and maternal relationship. Thank you, Maria.
Morning. I'm Maria. I'm a third-year PhD student, and today I will share with you a bit of my PhD project story. So first, I need to say that skin, as all you know, is our first barrier against the world. And in case of neonates, this skin plays a huge role in the protection of this neonate that came from the protective maternal uterus to the wild world. So when we born and at birth, we are colonized with a vast number of microorganisms, although Staphylococcus epidermidis is the major microorganism that colonize skin. This microorganism as well plays a huge role, an indispensable role in establish disease homeostasis in these babies, and like fighting the pathogens and induce immune response of these babies. So they help in educate the immune system of the babies. So this microorganism as well is seen as a harmless commensal microorganism, although now it starts to be seen as an opportunistic pathogen that can cause latent sepsis. So these babies are hospitalized, and most of the cases they need to have catheters to inject food or other type of analysis. So any skin breaches as catheters allowed these skin colonizers to enter in the microorganism and eventually can cause this sepsis. So in nearly 50% of the neonates that have pathogenic strains of epidermidis are responsible for the latent sepsis. So phages, we have phages in skin as well. So these phages in this environment are responsible for this host bacterial communities regulation. And specifically for the phages of that infect the epidermidis, specifically in neonate skin, little is known about that. All metagenomic studies have been done with these specific samples. So the main goal of my PhD is to understand this phage-bacterial relationship in this specific environment and understand if these phage therapies can prevent a neonatal sepsis. So first of all, we need phages. So for the phage isolation, we use 3 types of samples. So the first 2 samples came from babies, so these neonatal babies. So it was collected skin swab and feces from this baby every day since day 3 and day 7, and then every 2 weeks was taken a sample. And in addition, when these babies had sepsis, or at least suspect that they have sepsis. And in addition, it was take a skin swab from both parents. So we choose some samples and we mixed with the bacterial cocktail that was composed with 3 Staphylococcus epidermidis strains that came from skin. So 2 of them from adult skins, 1 from baby skin. We incubate them, take 2 time points, 4 hours and 24 hours, and then we run a spot test. So for all the samples, we analyze almost 500 samples, although only on 80 of these samples we have positive for the phage, which gives us 60% of success in recover phages. So in these 80 positive samples, we choose 37 using the criteria for at least 1 for baby, and when it's possible, one that came from a skin swab sample, a feces sample, and a parent sample. And in addition, we isolate three phages that came from sewage. And for this, we use 4 strains, all of them from skin, 2 from adults, and 2 from babies, which gives us a total of 40 phages. So with these 40 phages, we start to characterize them using observe the morphology, and then whole genome analysis and analytic spectrum. So starting for the morphology, we observed by transmission electron microscopy, and we found that we had 22 of podovirus-like phages, 14 cyphoviral-like phages, and 4 myovirus-like phages. Then we check the whole genome of these phages, and we check that all of these phages are lytic. And obviously, we can see the 3 clusters of the morphology that I talked to you about. And we understand as well that they are very similar between the same morphology. Then we had this data, and then we crossed with the data that we have from the samples that we isolated these phages. So, and analyze the homology between them. So we now have 4 cases that we have homology between the phages that are between 100 and 99% of homology. And these pairs of phages, 4 of them came from a mother and a baby. 2 of them came from the same baby, but different sample types. So one from skin, one for feces. And we have a case that they came from different babies, but we know that these babies were at the same time in the hospital. Then we have 2 cases. We have a pair of phages that have 98% to 97% of homology that came from same baby, but different sample type. And in this case, we have phages that are CFO and paired as a podo. And then we have homologies between 96% and 95% of homology. And we have a case that we have a pair mother-baby, and this case is a myovirus. And then same baby, different samples, we have 1 case. And then again, different babies, but in this case, we know that these babies are twins. Then we perform a lytic spectra using strains from 2 collections. So one of the collections that came from Portugal, so we call it SEP collection. And in this collection, we have 26 Staphylococcus epidermidis and more 6 coagulase negative staphylococci. And then 1 Staphylococcus positive, in this case 3 Staphylococcus aureus. And then came from the other collection, we have 153 Staphylococcus epidermidis. And these bacteria and these strains were isolated from the same samples that we try to isolate the phages. So in this assay, we use as well the phage concentrations between 110 to the 8 and 410 to the 8, so which gives us a total of 40 phage against 197 strains. So starting from the no S. epidermidis strains, we conclude that none of these phages could infect the 3 Staphylococcus aureus strains. Then, in relation to the podovirus-like phages, we conclude that only podo can infect and propagate in Staphylococcus equorum, hominis, and hemolytic strains. Relation to the CFO virus-like phages, only this CFO can infect and propagate in Staphylococcus werneri and Patencophery strains, and the myovirus-like phages only can infect and propagate in Staphylococcus werneri and Staphylococcus captis. Then when So the percentage of infection and propagation between SIFO and PODOS are very similar. Then when we talk about the S. epidermidis strains and compares from the different collection, we saw that in case of any of the podovirus like phage could infect and propagate in these strains. And SIFO virus and myovirus have a very similar percentage of infection and propagation. Then when we look in more detail for the collection that the samples came from the same samples as phages, we saw that the podovirus have the same behave, but the myovirus have a slightly percentage of infection and propagation. So the take-home message of the project right now is the isolation of this phage have a success rate of 16%. The prevalence morphotype is the podovirus-like phages, followed by SIFO virus-like phages and myovirus-like phages. In relation to the whole genome sequence, all are lytic phages and have a high percentage of homology between mother and baby samples and at the same baby. And the lytic spectra, in general, myovirus-like phages have a broad host range in Staphylococcus epidermidis strains in relation to the other morphotypes. So I like to acknowledge my agency, funding agency, obviously my supervisor, Dr. Luis Melo, Dr. Mercedes Agüero, and Professor Joana Azevedo, as well as the people from Azevedo Lab and Gomes Agüero Lab. And to conclude, I just talk a bit about one of my side projects. I have a podcast, and this podcast is about the phage world. So if you are interested in listen, you can get us on all the podcast platforms and follow us on X. We have more than 16 episodes with PhD students, early researchers, and well-known names in phage world. So if you can get us, you can get us through our email. So thank you for listening. I'm open to your questions.
Any questions for Maria?
That was a lovely talk. Thank you.
Thank you.
Did you find any? You said you were sort of wanting to see about the phages that might target the sepsis-inducing strains. Did you find strains that targeted? Phages that targeted
these strains? Yeah. Actually, we run a lot these lytic spectra against a lot of strains. We didn't check this pathogenic strain, like the difference between the pathogenics and commensal strains is a discussed thematic. So we run to different strains, and these strains have different genes that are associated with the pathogenic part of Staphylococcus epidermidis. So yeah, but I don't have conclusion about that.
Very interesting. You probably also have some resistant bacteria like MIG-A, MIG-C carrying strains. Do you see any correlation between phages and the carriage of the SCC-MIG resistant giving mobile genetic elements?
Yeah, it's very interesting, and it can lead with a conclusion that can be very useful. But actually, I don't check the bacteria. I run against a huge panel, but maybe it can be interesting to check in these bacteria if they have this. Thank you for your suggestion.